Assalamu alaikum everyone. In today's class, we will be going to discuss some difficult MCQs that appeared in AS level biology 9700 paper 1, and I will also be covering some new MCQs which are from the new syllabus, and some of these MCQs are taken from some other sources and some MCQs are made by me. So let's start with the class. We will go backwards from 40 to 1. MCQ number 40. Which graph shows the effect of carbon monoxide on the percentage saturation of hemoglobin with oxygen? Okay, so we have to choose a graph which shows the effect of carbon monoxide on the percentage saturation of hemoglobin with oxygen. Obviously, guys, as you all know, that carbon monoxide binds very strongly with hemoglobin. It binds very strongly with hemoglobin. So what happens? It prevents binding of oxygen. When carbon monoxide binds very strongly with hemoglobin, so carbon monoxide prevents the binding of oxygen to the hemoglobin. As a result, the percentage saturation of hemoglobin with oxygen will decrease. So in which curve do we see a decrease in the percentage saturation of hemoglobin with oxygen when carbon monoxide is present? So the only dotted line that shows decreased percentage saturation of hemoglobin with oxygen is found in option A. So option A is the answer. Other options show that either the percentage saturation of hemoglobin with oxygen stays the same or it increases. Guys, always remember that whenever carbon monoxide binds to hemoglobin, it binds to the heme group and prevents the binding of oxygen with the heme group. So the percentage saturation of hemoglobin with oxygen decreases. Let's move on to MCQ number 39. This is a very good MCQ. The stalk of a dandelion flower is a hollow tube. Pieces of stalk are cut as shown and placed in sucrose solutions of different water potentials. Okay guys, so what they have done over here is that they took a stalk of dandelion flower and they have cut the stalk into a strip, right? And this strip is placed in sucrose solutions of different concentrations. So what they have done, they have cut a strip which is somewhat like this. And guys, you should know that the strip that they have cut has two ends. One end has cells with thick outer cell walls, and the other end has cells with thin cell walls, and these cells are known as thinner walled inner cells, and thick walled outer cells are the cells which are outwards. Okay, so as you all know that the water will diffuse very slowly through the outer thick walled cells, and the water will diffuse very rapidly through the thin walled inner cells, right? So the MCQ says that which diagram shows the piece that is placed in the sucrose solution with the highest water potential. Guys, remember that if the strip is placed in a sucrose solution with highest water potential, that means that the water will go into the strip. So what do you think? From where the most water will go? Most water will go from the end where there are thin celled wall cells, right? So most water will enter from this side where there are cells with thin cell walls, and hardly any water will enter from this side. So what will happen is that, guys, the strip will be curved like this, because the end with cells with thin cell walls will expand more than the end with cells that have thick cell walls. So the strip will curve like this. The answer is A. I hope you get this MCQ. The strip curves because of the different rate of movement of water by osmosis. Or we can even say that the water does not cross the thick walled outer cells. So water enters from here and not from here. Let's move on to MCQ number 38. So guys, this MCQ is made by me. Let's discuss this one. This is regarding the introns and exons. Okay, MCQ number 38. A piece of DNA containing 8,000 nucleotides is transcribed and then translated. This piece of DNA had some introns interspersed between exons. When the piece of DNA was transcribed, the pre-messenger RNA was formed. The introns in the pre-mRNA had 1,000 nucleotides. What is the maximum number of amino acids found in the polypeptide chain? Okay guys, so in this MCQ, they say that there is a piece of DNA which contains how many nucleotides? This piece of DNA contains 8,000 nucleotides, which means that every strand contains how many nucleotides? 4,000 nucleotides. Okay. Then they say that this piece of DNA is transcribed. Okay. Before this point, they said that this DNA contains what? Introns interspersed between exons. So as you all know guys, that introns are what? Introns are non-coding regions and exons are coding regions. So they have mentioned that there are introns between the exons. For example, this is exon. So this segment is intron, and then this one is exon. Then this segment is intron, and this segment is exon, and this one is intron. So they have mentioned that this piece of DNA has introns interspersed between exons. And as you all know that introns are non-coding. Introns are non-coding regions of a gene. For example, if this is a gene, so introns are non-coding regions of the gene and exons are coding regions of the gene. Okay. Then they said that this DNA is transcribed to form messenger RNA. Obviously, messenger RNA, when messenger RNA is formed, messenger RNA is single-stranded. So it contains how many nucleotides? It contains 4,000 nucleotides because it is single-stranded. And they said that pre-mRNA was formed. Obviously, when transcription of eukaryotic DNA occurs, so first the pre-mRNA is formed, and this pre-mRNA will later be modified to form the final messenger RNA or final transcript. Okay. So because this is pre-mRNA and this is complementary copy of the template strand of DNA, so obviously it will also contain what? It will also contain exons and introns. Okay. So in the question they have mentioned that in the pre-messenger RNA, they're not talking about DNA. They're saying that in the pre-messenger RNA or pre-mRNA, 1,000 nucleotides consist of what? The introns, right? So introns are basically 1,000 nucleotides. So if introns contain 1,000 nucleotides, then what are the nucleotides? What is the number of nucleotides found in exon? Yes guys, it's 3,000. So 3,000 nucleotides are found in exons. So we can say exons contain how many nucleotides? 3,000 nucleotides. This means that 3,000 nucleotides are coding. Okay, guys, as we all know that 3 nucleotides code for one amino acid. So how many nucleotides are there which are actually coding? There are 3000 nucleotides which are actually coding. So what will be the number of amino acids in the polypeptide? The answer will be 1000 amino acids. So what is the maximum number of amino acids in this polypeptide that is formed by the transcription and translation of this piece of DNA? The answer is B, 1000 amino acids. Let's move on to MCQ number 37. The MCQ number 37 is similar but little bit different. A piece of DNA containing 12,000 nucleotides is transcribed and then translated. This piece of DNA had some introns interspersed between exons. All the introns altogether have 3000 nucleotides. Okay guys, so what is the difference between MCQ number 37 and MCQ number 38? They say that the piece of DNA has how many nucleotides? It has 12,000 nucleotides. And this DNA has introns and exons. Okay. So they say that in this DNA molecule, they haven't mentioned the pre-messenger RNA. They say that in this DNA molecule, all the introns together have 3000 nucleotides. So over here in the DNA, as we all know that intron is double-stranded, right? Okay. So they say that in the DNA, introns have 3000 nucleotides. This means if we talk about DNA that contains only exons, will be shorter and will have how many nucleotides? 12,000 minus 3000, because 3000 were introns and exons are how many? 9000. So there are 9000 exons in the piece of DNA. Okay, so for translation, what is formed? First transcription occurs and a messenger RNA is formed. And when this messenger RNA is formed, it contains how many nucleotides? It contains half nucleotides, because in the two strands there were 9000 nucleotides. So in one strand there will be 4500 nucleotides, right? So when the protein will be formed by translation, as we all know that 3 nucleotides code for one amino acid. So 4500 nucleotides will code for how many amino acids? The answer is 1500. So there will be 1500 amino acids in this polypeptide. So this is the method we can apply. There's another method that we can apply, is that as we all know that double-stranded introns contain 3000 nucleotides, right? So single-stranded introns in pre-mRNA could contain 1500 nucleotides. So what we can do is that if we go by this method, if we go like this, so for example, the DNA had how many nucleotides? 12,000. And when the pre-mRNA is formed, now how many nucleotides? 6,000. And out of all these nucleotides, 1500 will be introns. So the remaining will be 4500. Why 1500 and not 3000? Because we are talking about a single strand. The introns mentioned over here are in the DNA. If the examiner says that the number of introns are in the pre-messenger RNA, then that is the number for the pre-mRNA. If the examiner says in the DNA, then that is the number of nucleotides in the introns of the DNA. Okay, so I hope you get these MCQs. You can get these types of MCQs in your upcoming paper. Let's move on to MCQ number 36. This MCQ is also made by me. Which of the following is true about introns and exons? Okay, so introns are found only in eukaryotes. Guys, is this true? Yes. Always remember this point, that introns are only found in eukaryotes. However, exons are found in both eukaryotes and prokaryotes. Exons are basically coding regions, and prokaryotes only have exons. The gene of a prokaryote only contains coding region, and there are no non-coding regions in the gene of a prokaryote. However, eukaryotes contain both introns and exons. Introns are non-coding regions and exons are coding regions. Exons are only found in prokaryotes. No guys, they are also found in eukaryotes. Introns are coding regions of the gene. No, they are non-coding regions of the gene. Exons are coding regions of the gene. During splicing of pre-mRNA in eukaryotes, introns are removed and remaining exons are joined to form a final mRNA transcript. Guys, this statement is true, as you all know that the pre-messenger RNA contains both the introns and exons. So what happens? Splicing occurs. What is meant by splicing? Yes guys, splicing is basically a process by which pre-messenger RNA is modified in such a way that introns and exons are cut or separated. So there are splicing sites. What are these? These are splicing sites between the introns and exons. So there's a enzyme called spliceosome, and what spliceosome does is that spliceosome cuts the pre-messenger RNA at these sites which I have shown with the arrow. These are known as the splicing sites. What happens by splicing? By splicing by the spliceosome at splicing sites, introns and exons are separated, and what happens? The same spliceosome then joins the exons together. So the exons will be joined and the introns will be separated. This chain or transcript with only exons is known as final messenger RNA or mature messenger RNA, and then this mature messenger RNA is translated at ribosome. So this is true that during splicing, what happens? Introns are removed and exons are joined to form a final messenger RNA transcript. So the answer is 1 and 4, D. Okay, let's move on to MCQ number 35. Okay guys, let's move on to MCQ number 35. Portions of the DNA sequences of normal and mutant beta-globin genes are shown above. So guys, what is shown? There are portions of two DNA sequences. One is from the normal beta-globin gene, which codes for the normal beta-globin polypeptide. The other one is the mutant version or the mutant allele of the beta-globin gene. Basically, this is a portion. The lower one is the portion of the mutant allele of the beta-globin gene. The most plausible explanation for why the indicated mutation changing from A to G results in the disease beta-thalassemia in the mutation, or is that mutation. They say that the most plausible explanation for why the indicated mutation changing from A to G results in the disease beta-thalassemia is that the mutation changes the amino acid specified by the codon of which the altered bases part, generates a recognition site for restriction enzyme, so the gene is cut into two, creates a new splice site so that a portion of the intron is not removed, results in an increase in the transcription of beta-globin gene. So guys, what should be the answer? Let's look at this MCQ carefully. So there's a mutation, and where is the mutation? In the intron, right? There's a change from A to G. Okay. So guys, always remember this point, that whenever mutation occurs in the intron, always remember this point, that whenever mutation occurs in the intron is non-coding. So if a mutation occurs in the intron, so the amino acid will not change, right? So they say changes the amino acid specified by the codon, of which the altered base is the part. So if the codon changes, it does not code for any amino acid, it's an intron. So this could not be the option, right? Okay, generates a recognition site for restriction enzyme so that the gene is cut into two. Okay, so guys, over here we don't have any restriction site. Let me tell you what is a restriction site or recognition site for restriction enzyme. You will study in A2 that on the DNA there are some sites known as the restriction sites or the recognition sites for the restriction enzyme, where the restriction enzyme breaks the gene. So over here they haven't mentioned any restriction site, so this cannot be the answer. Okay guys, if you look at this point, what is this? As I just mentioned in the last MCQ, that between intron and exon there is a site known as splicing site, where splicing occurs, where the spliceosome cuts the pre-messenger RNA so that the introns and exons are separated. Over here we see a sequence that is TTAG. Can you see this? TTAG. And over here in the normal beta globin chain, in the normal beta globin gene, there was a sequence known as TTA, and this changed to TTAG. The sequence was TTAA, double T double A. Now the sequence is TTAG, which matches the sequence near the splicing site. So what has happened is that now the splicing will occur at this point. Are you getting my point? So what will happen? The splicing will occur at this point instead of this point. The splicing will occur at some point before. And because of that, what will happen? That intron will be included in the exon. So there will be some extra bases which will become coding. And these will code for extra amino acids, resulting in a non-functional polypeptide, right? And this will cause a disease called beta-thalassemia, because this will result in the formation of an abnormal beta globin polypeptide chain. So the answer should be that this mutation creates a new splice site so that the portion of the intron is not removed. The answer is C. Let's move on to MCQ number 34. Which relationships could be investigated using a colorimeter? So guys, always remember that colorimeter is a device which records the intensity of the color of a solution. I'm repeating it again. What is the colorimeter? Colorimeter is simply a device that records the intensity of color of a solution. What colorimeter does is that how does the colorimeter records the intensity of the color? It does this by recording the absorption of light by the colored solution, right? Colorimeter measures how much light is absorbed by the colored solution. So higher the intensity of the light, higher the intensity of the color, higher the absorption of the light, right? Let me repeat it again so that it becomes clear in your mind, that colorimeter is a device which measures the intensity of the color of a solution. So how does the colorimeter measures the intensity of the color? It measures the intensity of the color by measuring the absorbance of light that the color has absorbed. Obviously, darker the color, the more the intensity of the color, the more will be the absorption of light by the color, because as we all know, the darker colors absorb more light than the lighter colors. So always remember this point, that higher the absorbance of light, darker will be the color, right? Okay, so as I just mentioned, that colorimeter measures the intensity of the color in a solution, or measure the absorbance of light by a solution. So guys, we can always place solution in the colorimeter. We can place the test tube containing solution in a colorimeter. Okay, so which relationships they are asking that which relationships could be investigated using a colorimeter. The effect of light intensity on the rate at which a solution of light sensitive dye changes from green to colorless. So guys, we can investigate this relationship by colorimeter because in this reaction, what are we observing? That there is a light sensitive dye which is changing color from green to colorless. So this can be investigated using a colorimeter because there is color change and change in the intensity of the color can be recorded by the colorimeter. The effect of temperature on the rate of breakdown of cell membranes in tissues with pigmented cells such as the beetroot red beet. Guys, this relationship can also be investigated using a colorimeter. And what is the reason? Because as we increase the temperature, more cell membranes will be broken down and more pigment will be released from the cells. So as the pigment is releasing, pigment is red in color, and this can be the absorbance by this color can be measured by the colorimeter. So this relationship can also be tested with colorimeter. The effect of pH on the rate of release of oxygen from the breakdown of hydrogen peroxide by catalase. Guys, this relationship cannot be tested using a colorimeter because there is no color change and no colors are involved. Over here, oxygen gas is being released, which is colorless gas. Okay. Let's move on to option 4. The effect of light intensity on the rate of change of skin color of lizards that become paler in bright light. Guys, this relationship cannot be tested using a colorimeter because they are talking about the skin color of lizards. We can only deal with solutions using a colorimeter. We cannot obviously put the lizards in a... Can we put the lizard in a colorimeter device? No, obviously no. We cannot put the lizard into a colorimeter device. Obviously we cannot do that. We can simply put the test tube containing the solution in the colorimeter device. Okay, so option 4 is wrong. The answer will be 1 and 2, that is B. Let's move on to MCQ number 33. Which disease is not likely to be passed directly from parents to child? So guys, remember that if we talk about cholera, so cholera can be passed from parents to child directly because if a parent cooks, if one of the parent has cholera and if one of the parent does not wash hands properly and use those hands to cook the food at home, and if the child eats that food, the cholera can be transmitted. So cholera is likely to be transmitted or passed from parents to children directly. If we talk about malaria, so guys, malaria is the disease that cannot be passed directly from parents to child. Guys, malaria can be passed from parents to child but not directly. There should be a female Anopheles mosquito in between because the female Anopheles mosquito will bite the infected parent and will take the plasmodium and then bite the child to transmit malaria. So there must be a female Anopheles mosquito between parents and the child to transmit malaria. Parents can transfer or pass malaria to the child but not directly. So the answer is malaria. If we talk about sickle cell anemia, guys, sickle cell anemia can be passed from parents to child because it's a genetic disorder which can be inherited. And if we talk about tuberculosis can also be passed from parents to child. That's because tuberculosis spreads via respiratory droplets. So if child is in close contact with the parents, the TB can easily be transmitted. Let's move on to MCQ number 32. Okay, the mechanism of action of 4 drugs that inhibit DNA replication is stated below. So guys, there are 4 drugs that inhibit DNA replication and the mechanism of action of these drugs are mentioned below. So there is one drug known as acetylcholine that inhibits DNA polymerase. Okay, there is another drug known as cytarabine that is converted into a molecule that can substitute for a DNA nucleotide and also inhibits DNA repair mechanisms. So guys, cytarabine is basically a drug that can be converted into a molecule that can substitute for a DNA nucleotide. For example, it can come instead of thymine nucleotide or adenine nucleotide or guanine nucleotide. It's basically a substitute for the DNA nucleotide, and cytarabine also inhibits the repairing of DNA. Right. The third drug is epirubicin that inhibits an enzyme involved in the unwinding of the DNA and separation of strands. So guys, which enzyme is involved in the unwinding of DNA or separation of DNA strands? The enzyme is DNA helicase. So guys, this enzyme is inhibited by epirubicin. Okay. The last drug is hydroxycarbamide. This inhibits an enzyme involved in the production of deoxyribonucleotides, which are the nucleotides used to synthesize DNA. So guys, hydroxycarbamide will inhibit the synthesis of the nucleotides, the deoxyribonucleotides, which are actually the nucleotides that are used to make DNA, and these nucleotides are required for DNA replication. Okay, which row correctly matches a drug to an explanation of the mechanism of action? Okay, decreased pool of available nucleotides inhibit chain elongation. So guys, they say decreased pool of available nucleotides, so there will be less nucleotides. As a result, the chain elongation or the DNA elongation will not occur. So which drug will cause the decreased pool of available nucleotides? The answer is hydroxycarbamide. That's because we just read that hydroxycarbamide, what it does, it inhibits an enzyme that is involved in the production of deoxyribonucleotides. So there will be decreased pool of available nucleotides for the chain elongation. Okay, DNA strands not available as templates for transcription. So guys, if DNA strands are not available as templates for transcription, it means that the DNA templates cannot separate. DNA templates can, it means DNA strands cannot separate, sorry, right? When the DNA strands separate, DNA is double stranded as you all know. When the DNA strands separate, then the DNA strands act as templates, right? If the DNA strands are not available as templates, it means that the DNA strands cannot separate. And that is done by which drug? Epirubicin. That's because epirubicin inhibits DNA helicase, which causes the unwinding of DNA. So if the DNA does not unwind, then the DNA strands will not be available as templates. DNA damaged during replication and cell death occurs. So guys, as we just read, that cytarabine is a drug which inhibits DNA repair mechanisms. So DNA will be damaged, and DNA damaged during replication and cell death occurs, right? So this is done by cytarabine, because cytarabine inhibits repair mechanisms and also form a molecule that can substitute for DNA nucleotide. So cytarabine will first damage the DNA, and then it will also inhibit the DNA repair mechanisms. Exposed DNA template strands unable to be copied. That's because when the exposed DNA template strands, when the template strands are there but they are unable to be copied, that's because DNA polymerase is not working, because DNA polymerase is the enzyme which actually copies the DNA. It joins the nucleotides which are copying the strand. So DNA polymerase actually copies the strand. So acetylcholine will inhibit DNA polymerase, and it will cause the exposed DNA template strands unable to be copied. Let's move on to MCQ number 31. The diagram shows how genetically identical frogs can be developed from unfertilized frog eggs. The diploid number 2n for the frog is 26. Okay, so guys, in this MCQ, they are saying that genetically identical frogs can be developed from unfertilized frog eggs. So there will be frog eggs which come from the female frog, and from these unfertilized eggs, these will not be fertilized by the sperm. These unfertilized eggs will now be used to make genetically identical frogs. Okay, so they say that the diploid number 2n for the frog is 26. So if we talk about V, which is the unfertilized frog egg, eggs are gametes as you all know, and gametes are formed by meiosis. And because gametes are formed by meiosis, gametes are haploid, so they have half the number of diploid chromosomes, right? They have half the diploid number. So if diploid number is 26, that means the chromosomes in V will be 13. Okay, so what happens over here is that the nucleus is destroyed by the ultraviolet radiation. As a result, the nucleus is lost. A cell is taken from the frog. So guys, this is the normal diploid cell that is taken from the frog. This will contain 26 chromosomes, and the nucleus from the normal cell is inserted into the unfertilized egg from which the nucleus was initially removed. So this cell will also contain how many chromosomes? 26 chromosomes. So this X develops into a tadpole, and tadpole develops into a frog, and this frog will be genetically identical to the original frog. Okay. Which combination of numbers correctly identifies the number of chromosomes in each of the types of cells in the diagram? Okay, so guys, as we just figured out that V contains 13 chromosomes, and if we talk about X contains 26 chromosomes, and W also contains 26 chromosomes. The answer is C. Let's move on to MCQ number 30. The diagram is a drawing from an electron micrograph of a typical animal cell. So this is a drawing of an animal cell from a picture taken from electron micrograph, or picture taken from electron microscope, which is known as electron micrograph. Okay. How many layers of phospholipids will a molecule of oxygen cross to travel directly from X outside the cell to Y, the matrix of mitochondrion? So guys, if we talk about the movement of oxygen molecule from X till Y, so how many membranes does this molecule crosses? It crosses how many membranes? 1, that is the cell surface membrane, 2, that is the outer membrane of the mitochondrion, and 3, that is the inner membrane of the mitochondrion, and then it reaches the Y. So it crosses 3 membranes. And as you all know that each membrane is a phospholipid bilayer. Each membrane contains 2 layers of phospholipids, and they have said how many layers of phospholipids. Guys, they haven't mentioned the phospholipid bilayer. They have mentioned how many layers of phospholipid. So because the oxygen molecule is crossing 3 membranes or 3 phospholipid bilayers, it means the oxygen is crossing 6 phospholipid layers, because 3 phospholipid bilayers will be equal to 6 phospholipid layers. The answer is D. Let's move on to MCQ number 29. One type of congenital heart defect is called the atrial septal defect. So guys, what is meant by congenital heart defect? A heart defect that is present from birth. Okay, so one type of congenital heart defect is called the atrial septal defect, where the left and the right atria are not completely separated. Guys, this means that there's a hole in the wall or in the septum between the left and the right atria. So right atria and left atria are separated by interatrial septum, which is the septum between the 2 atria. So when they are not completely separated, there's usually a hole, and that is known as the atrial septal defect. So what will happen? ASD usually results in the blood moving from left atrium into the right atrium. So what will happen, guys, if there are 2 chambers over here? One is the right atrium and the other one is the left atrium. So what will happen? There's a hole in the wall known as the atrial septal defect. So what will happen? The blood will travel from left atrium to the right atrium. And why is that so? Because the blood pressure in the left atrium is higher than the blood pressure in the right atrium. So there will be left to right shunt, right? So they say over here that ASD usually results in blood moving from left atrium into the right atrium. This will cause increased blood pressure in the right atrium and decreased blood pressure in the left atrium. Because initially blood pressure will be higher in left atrium and lower in right atrium, but as the blood moves from left atrium to right atrium, the blood pressure in the right atrium will increase. And obviously, if the blood pressure in the right atrium will increase, this blood will go into the right ventricle, so blood pressure in the right ventricle will also increase, and the blood from the right ventricle will go into the pulmonary artery, so the blood pressure in the pulmonary artery will also increase. Okay, so they ask that which row describes the other effects of ASD on blood pressure and oxygenation. So they are asking you about the other effects of ASD on blood pressure and oxygenation. So guys, if we talk about the blood pressure in the pulmonary artery, it will also increase. As I just mentioned, that if the pressure in the right atrium increases, because right atrium is overloaded with blood, right atrium is receiving its own blood from the vena cava. Right atrium is receiving its own blood from the superior and inferior vena cava, and it is also receiving blood now from the left atrium. So right atrium is overloaded with the blood, and so is the right ventricle, and so is the pulmonary artery. So when the pulmonary artery is overloaded with blood pressure in the pulmonary artery increases. If we talk about aorta, the blood pressure in the aorta will decrease. And why is that so? Because as the blood from left atrium is going into the right atrium, less blood goes into the left ventricle. When less blood goes into the left ventricle, less blood goes into the aorta. And if the less blood goes into the aorta, the blood pressure in the aorta decreases. Guys, if we talk about the percentage oxygenation of blood in the pulmonary artery, so as you all know that pulmonary artery receives deoxygenated blood from the right ventricle. You all know that right ventricle pumps blood into the pulmonary artery. So normally pulmonary artery has a deoxygenated blood, but over here we can see that blood is moving from the left atrium to the right atrium. So this blood is oxygenated, and this oxygenated blood goes into the right ventricle and then pulmonary artery. So percentage oxygenation of blood in the pulmonary artery increases. Percentage oxygenation of the blood in the pulmonary artery increases because the blood is now oxygenated. So the answer is D. Let's move on to MCQ number 28. Okay. Which definitions of diploid and haploid are true for typical eukaryotic cells? Okay. So guys, there are two definitions of diploid and two definitions of haploid. We have to state that which definitions for diploid and haploid cells are correct. Okay. So let me guide you regarding the diploid and haploid because many students are weak in this concept. Guys, if we talk about diploid cell, so what is a diploid cell? Diploid cell is a cell which contains two sets of chromosomes. How many sets? Two sets of chromosomes. What does that mean? Is that diploid cell contains one set from the father and one set from the mother. So the set from the father is known as the paternal set and set from the mother is known as the maternal set. So because the cell has two sets, it's known as diploid. If we talk about haploid, so haploid cell contains single set of chromosomes. Haploid cell contains single set of chromosomes. There's only one set. Okay, so guys, our normal body cells are diploid. If I talk about my liver cell, if I talk about my brain cell, all of these cells are diploid. All of my cells which are diploid contain half chromosomes from my father and half chromosomes from my mother. I got one set from my father and one set from my mother. But the gametes that I'm producing, for example, I'm male and I'm producing sperm. So my gamete will contain only one set of chromosomes, because as you all know that the haploid cells have half the number of chromosomes as compared to the diploid cell. For example, if diploid cell has 46 chromosomes, then the haploid cell will have 23 chromosomes, as you all know. So diploid is represented by 2n and haploid is represented by n, where n is the number of chromosomes in one set, n is the number of chromosomes in one set, n is number of chromosomes in one set. Okay, so if you want to figure out the number of chromosomes in the haploid cell, you just need to know the number of chromosomes in one set. If you need to know the diploid number of a cell, then you need to know the number of chromosomes in one set, and if you know the number, you can multiply it by 2 to get the diploid number. For example, over here haploid number is 23. That is the number of chromosomes in one set. Then the diploid number will be 23 into 2, that is 46. So I hope you get this basic point. The diploid cells contain two sets of chromosomes, one from the father and one from the mother, and haploid cell contains a single set of chromosomes. And haploid cells are always gametes. The normal body cells are diploid and the gametes are haploid. And this is the what is the reason of gametes being haploid? Because as I just mentioned that we get one set from the father and one set from the mother. So to transfer one set, the father transfers us the sperm, and to transfer one set of chromosomes, the mother transfers us the ovum. I hope you get it now, that why haploid cells must contain one set, because these are the gametes. They come from both the father and the mother. So when they fuse, when the gametes fuse, when the haploid gametes fuse, they form the diploid cell known as the zygote, and from the zygote we then develop. Okay, one more thing I would like to tell you about the diploid cell is that, guys, the diploid cell has homologous chromosomes. So guys, how do we define homologous chromosomes? If in a cell you see two chromosomes of same size and shape, if in a cell you see two identical chromosomes like this, and also these one, and also these. So guys, we have three pairs. One is this pair of identical chromosomes, one is this pair of identical chromosomes, and one is this pair of identical chromosomes. So guys, if you see identical chromosomes in a cell, then that cell is diploid. These identical chromosomes are known as homologous chromosomes. So guys, this pair is known as the homologous chromosomes. So in the diploid cell, you will always find homologous chromosomes. This is how you recognize a diploid cell. So if we talk about this diploid cell will be 2n obviously. And what is the diploid number over here? There are total 6 chromosomes. So we can say 2n is equals to 6. This is a diploid cell which contains 6 chromosomes. If we talk about chromosomes in one set, so in the chromosomes in the one set will be 3 and chromosomes in two sets will be 6, right? This is the feature of diploid cell. If we talk about haploid cell, guys, always remember this point, that haploid cell does not contain homologous chromosomes. Haploid cell contains only one copy of each homologous chromosome. So for example, if we make the cell haploid, so there will only be one copy of each homologous chromosome. There will be no pairing, there will be no pairs, there will be no homologous chromosomes, only one copy of each homologous chromosome, right? So in a cell, if you don't see identical chromosomes, if you don't see any pair of identical chromosomes, it means that cell is haploid. So we can say this is n, this is haploid, and over here n is equals to 3. Okay, now let's discuss this MCQ. I hope we have discussed enough about diploid and haploid cell, and now we have enough information to solve this MCQ. Okay, so they say that which definitions of diploid and haploid are used for typical eukaryotic cells. So we have to select the correct definitions for diploid and haploid. Diploid, any cell with an even number of homologous chromosomes. So guys, this statement says that any cell with an even number of homologous chromosomes. Guys, how many homologous chromosomes are there in a diploid cell? Always remember this point, that in a diploid cell there are only two homologous chromosomes, right? They have mentioned even number of homologous chromosomes. This is wrong, because even could be 4, even could be 6 as well, even could be 8, right? So there should be that any cell with 2 homologous chromosomes, right? Homologous chromosomes should be 2. Each type of homologous chromosomes should be 2, or we can say 2 copies of each homologous chromosomes, right? So this statement is wrong. Diploid, any cell with 2 copies of each homologous chromosome. This is the correct statement, guys. This is so true. As I just mentioned, the diploid cell is any cell with 2 copies of each homologous chromosome. For each homologous chromosome, we have 2 copies in a diploid cell. There cannot be even number of copies. Even can be 2, 4, 6. So that's why statement 1 is wrong. Haploid, any cell with half the diploid number of homologous chromosomes. Guys, diploid number is not for the homologous chromosomes. Always remember this point, that diploid number is for the cell. Diploid number is not for the homologous chromosomes. We can see that there are 2 homologous chromosomes, but the diploid number is 6. Diploid number is not for the homologous chromosomes. Diploid number is for the cell. So statement 3 is wrong. Haploid, any cell with 1 copy of each homologous chromosome. Yes guys, this is true, that there is only 1 copy of each homologous chromosome. So the answer should be D, 2 and 4. Okay guys, let's move on to MCQ number 27. Lipase is a digestive enzyme produced by the pancreas that catalyzes the hydrolysis of dietary lipids. The table shows how the pH of a liquid food containing a high proportion of liquids decreases over time. So guys, in this experiment, what do we see? They have given us the information that lipase is a digestive enzyme that is produced by the pancreas, and what lipase does is that lipase causes the hydrolysis or breakdown of lipids. And we can see that there was a liquid food that contains high proportions of liquids, and when this liquid food was being acted upon by the lipase, so lipase breaks down these lipids and the pH falls. So guys, can you tell me why the pH falls when the lipase is acting on the lipids? Obviously, when lipase acts on the lipids are broken down into fatty acids and glycerol, and the fatty acids, they decrease the pH. So what do we see in the graph is that initially the pH is decreasing, and then the pH becomes, yes guys, from 50 to 60 minutes the pH becomes constant. So the pH stops decreasing. This means that fatty acids are no longer produced and reaction has stopped, because the pH was falling because of the production of fatty acids, as fatty acids are acidic. So this means that from 50 to 60 minutes the fatty acids are not being formed and the reaction has stopped completely. Okay, so what could be the reason for the reaction to stop? Let's have a look. Which statements are possible explanations of the results of the experiment between 50 and 60 minutes? So guys, we need to take the correct statement for the possible explanation for the result that is between 50 to 60 minutes. That means the possible explanation for the reaction to stop. Enzyme concentration becomes a limiting factor. You guys always remember this point, that if the enzymes become limiting, this means that as compared to substrates, we have less enzymes. So it doesn't matter guys, even if enzymes are limiting, the substrates are a lot in number. This means that eventually more products will be formed and the reaction will not stop. Reaction will not stop if the enzyme concentration is limiting, right? Reaction stops when substrate concentration is limiting, or when the substrate decreases and finishes, and at that point the reaction stops. The limitation of enzyme concentration will never stop the reaction. The reaction will still continue because substrate is there and enzymes can be reused, as you all know. Substrate concentration becomes a limiting factor. Yes guys, if the substrate concentration becomes limiting, if substrate concentration decreases and if the substrate finishes, the reaction will stop and there will be no change in the pH of the liquid food, as the lipids will not be broken down into fatty acids. All the enzyme active sites are saturated. Guys, the statement 3 and statement 1 are same. When the enzyme concentration becomes limiting, what happens? Substrate is far greater than the enzyme, so all the active sites of the enzymes are filled, all the enzymes are saturated, and the remaining substrates are waiting for their turn. So enzyme 1 and enzyme 3, option 1 and option 3 are the same statements, so they both are wrong. Denaturation of enzyme by the products. Guys, this could be a correct explanation, because as we all know that the lipids are broken down and the product that is formed is fatty acids, and fatty acids, they are decreasing the pH of the liquid food, and this fall in pH can denature the lipase. So denaturation of lipase or enzyme by the products, this could be the correct explanation, because fall in pH can denature the lipase. Okay, products are acting as competitive inhibitors. Yes guys, products can act as inhibitors of the enzyme. The products that are being formed can inhibit the enzyme and stop the reaction. So the answer will be 2, 4 and 5. The answer is D. Let's move on to MCQ number 26. The diagram shows apparatus set up to investigate the effect of putting a 10% glucose solution in a selectively permeable membrane bag, Viscine tubing, and surrounding it with water. So guys, if we look at this diagram, what they have done, there is a bag known as the Viscine tubing, and the wall of the Viscine tubing is partially permeable. Inside the bag there is 10% glucose solution, and outside the bag in test tube there is water. So guys, what will happen, as you all know that water has a higher water potential, and 10% glucose solution has a lower water potential, so water will enter the whisking tubing by osmosis. And as you all know, what will happen to glucose? Glucose can cross the partially permeable membrane of whisking tubing, so glucose will escape out into the test tube of water. Okay, samples from the surrounding water were tested with Beta solution after 10 minutes and after 20 minutes. The change in the volume of glucose solution was observed after 20 minutes. So guys, what they are doing, that they are observing the change in the volume of water, and they are also taking out the water in the test tube to check it with Benedicts for reducing sugars. So obviously, as we all know, that glucose is coming out of the whisking tubing into the water, so water will now contain the reducing sugars. So result with the Benedicts test will be positive. After 10 minutes there will be little glucose in the water, so the color with Benedicts will be green, and after 20 minutes there will be a lot of glucose in water, so the color will become darker and it will become orange. And as we all know, that the volume of the glucose solution in the whisking tubing will increase. Why the volume of the glucose solution will increase? Because water is entering into the whisking tubing. The answer is B. Let's move on to MCQ number 25. A person's blood group is determined by antigens present on the red blood cells. Okay, so they say that the blood group of a person is determined by the antigens that are present on the red blood cells. The table shows the antigens and antibodies in the blood of people with different blood groups. So guys, you should know that if we talk about the red blood cells, so red blood cells of different blood groups have different antigens on their cell surface. So guys, if we talk about the red blood cell of an individual with blood group A, there will be antigen known as A antigen. So guys, if blood group is A, the person contains antigen A. If blood group is A, the antigen on the red blood cell will be antigen A. If the blood group is B, then the antigen on the red blood cell will be antigen B. If the blood group is AB, then there will be both A antigens and B antigens on the surface of the red blood cells. And if the blood group is O, then guys, there will be no antigen on the surface of the red blood cell. The red blood cell looks like an O, that's why O blood group. I hope you get this point. Okay, so in this MCQ, they say that if a person has a blood group A, the person will have an antibody against the blood group B. Always remember this point, that if you have an antigen, and if that antigen is your antigen, self antigen, not foreign. Always remember this point, guys, that if the antigen is yours, if it's your body's antigen, if it's self antigen, antibody will never be produced against self antigen. Antibodies are produced against foreign antigens. So if any person has blood group A, the person will have A antigen and will have antibody against B antigen, because for the person with blood group A, the foreign antigen will be antigen B. So the antibody will be against blood group B. If a person has blood group B, so the person will not have the antibody against the B. Instead, the person will have antibody against A, which is known as anti-A antibody. If a person has blood group AB, so the person will not have any antibody, because both A and B antigens are self for the person. So the person will not make any antibody against antigen A or antigen B. So there will be no antibodies against any antigen in the person with blood group AB. Because the person with blood group O has no antigen, so the person with blood group O, because the person has no antigen, so it will contain antibodies against both the antigens A and B. So this is anti-A antibody and anti-B antibody. What is the reason of both the antibodies present in the blood group O? Because the person with blood group O has none of the antigens A or B. So for the person with blood group O, antigens A and B are foreign. So antibodies will be formed against antigen A and antigen B. That's what is mentioned in the table, that if a person has blood group A, the person has A antigen and anti-B antibody. If the blood group is B, the person has B antigen and anti-A antibody. If blood group is AB, then the person has A and B antigen and neither of the antibody. If the blood group is O, the person has no antigen and both anti-A and anti-B antibody. So they say during a blood transfusion, they are asking that during a blood transfusion, it is essential that the recipient's blood does not contain antibodies to the donor's blood. What are they saying? That during transfusion of blood, what is necessary? That the recipient which is receiving the blood does not contain any antibody against the donor blood. Which blood groups can be given to a person with blood group B? So if a person has blood group B, if the person has blood group B, then the person definitely has anti-A antibody. So any blood group with A antigen should not be given. So A cannot be given, guys. AB cannot be given. So the answer should be D. If a person has blood group B, so the blood that should be transfused should also be blood group B, because that blood will not be rejected because the antibody is anti-A in the people with blood group B. Okay. And if we talk about O, so O has no antigen. So this person can receive this blood group because anti-A antibody will not be effective against the O blood group. That's why the answer is D. Let's move on to MCQ number 24. The graph shows the changes that take place in the volume of the left ventricle during one cardiac cycle. Which point on the graph represents the start of the atrial systole? Okay guys, so this is a graph on which on y-axis there is ventricular volume. So guys, this is the volume of the left ventricle during the cardiac cycle, right? Okay, so they are asking that which point on the graph represents the start of the atrial systole. So guys, as you all know that when the atria contract, or for example when the left atrium contracts, what will happen? When the left atrium contracts, the blood will be squeezed into the left ventricle, so the volume of the left ventricle will increase. So over here there are two options that can be the answers, B or C, because they say start of the atrial systole. Because as the atrial systole starts, after that the volume of the ventricle should increase. So the volume of the ventricle is increasing after B and after C. So what do you think? What should be the answer? Guys, always remember this point, that whenever the atrium contracts, whenever the left atrium contracts, it pushes the blood into the ventricles. It pushes the blood into the left ventricle. So the left ventricle volume definitely increases, but there will be a mild increase. Why? Because the left atrium contracts to squeeze the remaining blood. Left atrium contracts. Why does the left atrium contracts? It contracts to squeeze the remaining blood in the left atrium into the left ventricle. What is meant by the remaining blood? Guys, what happens during diastole is that both the left ventricle and left atrium are relaxing. What happens during diastole? The entire heart is relaxing, and diastole is known as the filling phase of the heart. So during diastole, what happens is that the left ventricle is relaxing and the left atrium is also relaxing, and much of the blood trickles from the left atrium to left ventricle during diastole. Guys, you should know that what happens during diastole, that much of the blood from the left atrium trickles down into the left ventricle during diastole when the heart is relaxing. Only the remaining blood, which was unable to trickle into the left ventricle, is squeezed by the left atrium during systole. Guys, always remember this point, that most of the blood from the left atrium to left ventricle goes in diastole. This is known as passive filling. There is no energy required. There is no active process. Most of the blood during cardiac cycle flows from the left atrium into the left ventricle during diastole by passive filling. The blood that remains in the left atrium, the small blood that remains in the left atrium, is pushed by the atrial systole. Right? The reason for pushing is to eliminate the remaining blood. Always remember this point. So the answer should be C, because after C we can see, because just right after C there is only mild increase in the ventricular volume. At B there is a major increase. After B there is a major increase in the ventricular volume. So guys, after B is basically what? Diastole. Because after B, what happens? There is passive filling. Okay, guys, if we talk about A is ventricular systole, because the ventricular volume is decreasing and the ventricle is emptying. This means ventricle is contracting. Ventricular systole. And C is atrial systole. Okay, let's move on to MCQ number 23. The diagram shows the results of an experiment using leaves with the same surface area from the two different species of plant. Each leaf was left on a balance in daylight in a closed room, and its mass is recorded at 1 hour intervals. So guys, what are they doing in this experiment? They have taken two leaves, and the two leaves have same surface area. However, they are from the two different species of the plant. Each leaf was left on balance in the daylight. So each leaf was left on a balance. That was actually electronic balance, which was measuring the mass. So each leaf was left on a balance in daylight in a closed room, and its mass is recorded at 1 hour intervals. So we can see that the mass of the leaf is falling. This is because of the transpiration, guys. As we all know that water vapor is lost from the leaf, and this process is known as transpiration, and because of the transpiration the mass of the leaf falls. So we can see that both the leaves have the same surface area. However, the leaf 2 loses more mass. It means there is higher rate of transpiration in leaf 2. There is higher rate of transpiration in leaf 2. That's why the leaf 2 is losing more mass, and there is lower rate of transpiration in leaf 1. Okay, which features of leaf 2 could explain these results? So guys, we have to state the features of leaf 2 which could explain these results, and the results are that leaf 2 has a higher rate of transpiration. So what could be the features of leaf 2 that could lead to the high rate of transpiration? More stomata per unit area of the leaf. Yes guys, if there will be more stomata per unit area of the leaf, if there will be more stomata per unit area of the leaf, the rate of transpiration will be higher, and this is true. Fewer trichomes, hairs on the leaf. Yes guys, if there will be fewer hairs or trichomes on the leaf, so less water vapor will be trapped, and there will be less humidity around the leaf and around the stomata, so more water vapor will be lost. As you all know that trichomes are the features of xerophytes, and what trichomes do is that trichomes trap water vapor and create a humid region around the stomata, so that the humidity increases and the rate of transpiration decreases. But over here, if the leaf has the fewer trichomes, it means less water vapor will be trapped and less humidity will be created. So as you all know that when the humidity decreases, when there is less humidity around the stomata, so the rate of transpiration increases. Sunken stomata, guys, this is wrong. Sunken stomata do that they decrease the rate of transpiration, and if leaf 2 had sunken stomata, the rate of transpiration would be lower in leaf 2. Okay, if we talk about thinner cuticle, then as you all know that thinner the cuticle, more the loss of water vapor. So the thinner cuticle will increase the rate of transpiration. So the answer should be 1, 2 and 4. The answer is B. Let's move on to MCQ number 22. two experiments were carried out using an enzyme from humans. The first experiment X was carried out at a constant temperature of 37 degrees Celsius. During the second experiment, the temperature was increased from 37 degrees Celsius to 80 degrees Celsius. All the factors were kept the same. Which graph shows the results? So guys, in this MCQ, what they are doing, that there are two experiments. Two experiments were carried out using an enzyme from humans. So they took the enzyme from the humans and they carried out two experiments. So one experiment that is known as the experiment X. It was the first experiment. It was carried out at a normal temperature and the temperature was constant to 37 degrees Celsius. So the experiment X, or the first experiment, was carried out at 37 degrees Celsius and the temperature was constant throughout. Okay. And the second experiment, what they did is that they started the experiment, and during the experiment what they did is that they increased the temperature from 37 degrees Celsius to 80 degrees Celsius, which means that the enzyme will eventually denature. All the other factors were kept the same. Which graph shows the results? So guys, if we talk about the second experiment, in the second experiment what they have done, that they started the reaction, and then during the reaction they started increasing the temperature from 37 to 80, causing the enzymes to denature. So what will happen, that initially the reaction will be rapid and the products will be formed, but as the enzymes denature, the reaction will stop and the product formation will stop. So there will be a lower product concentration as compared to the first experiment, because the reaction stops earlier. The reaction stops quickly without converting all the substrates to product. That's because the enzymes denature, and the enzymes denature due to increase in temperature. So the answer should be A. The answer cannot be B, because guys, the product concentration cannot fall like this. The product concentration cannot fall after the enzymes are denatured. The product concentration will become constant because no more product will be formed after the denaturation of enzymes. There is product concentration on y-axis, so you should note this point. Okay guys, when you increase the temperature from 37 to 80, the product concentration of the second experiment will not be equal to the first experiment. This is wrong. Product concentration will be lower than the first experiment. And in the option D, they are showing that product concentration was highest in the start and then it is falling. This is not true. The answer is A. Okay guys, let's move on to MCQ number 21. Which of these statements explain why some vaccines can be taken by mouth but tuberculosis vaccine has to be injected? Okay guys, they say that which statements out of these three would explain that why do we need to inject TB vaccine rather than taking it by mouth. As you all know that some vaccines can be taken by mouth, like the oral polio vaccine and oral cholera vaccine, right? That vaccine can be taken by the mouth, but TB vaccine can only be injected. It cannot be taken by the mouth. What could be the right explanations? Let's see. Macrophages present antigens in the vaccines to stimulate an immune response. Okay guys, it's fine. You all know that this is true, that whenever an antigen enters the body, so macrophages present that antigen to the B and T lymphocytes to stimulate immune response. But if we take the vaccine by mouth, still the macrophages would present this antigen to the B lymphocytes and T lymphocytes, because the stomach wall contains a lymphoid tissue that contains macrophages. So macrophages are found in the stomach wall, because macrophages, as you all know, are found in tissues. So even if we take TB vaccine by mouth, if the antigen comes in contact with the macrophages, they will definitely present. So this could not be the correct statement, because we have to state the reason why we cannot take TB vaccine by mouth, right? So this cannot be the reason why we should not take the TB vaccine by mouth. This should not be the reason, right? Because even if we take the TB vaccine by mouth, if the antigen in the TB vaccine come in contact with the macrophages, they will definitely present the antigen to the B lymphocytes and T lymphocytes. The TB antigens necessary to produce an immune response. The TB antigens that are necessary to produce an immune response are proteins which would be digested in the stomach and small intestine. Yes, this is the perfect explanation, because as you all know that TB vaccine contains TB antigens, which are proteins in nature. And if you take TB vaccine orally by mouth, so what will happen? The antigens of the TB vaccine will be digested by the proteases in the stomach and the small intestine. So the vaccine will become ineffective. There are no B lymphocytes and T lymphocytes in the stomach. Guys, in the wall of the stomach, there is a lymphoid tissue which contains B lymphocyte and T lymphocyte. So this is not true. B lymphocytes and T lymphocytes are found in the stomach. The only correct statement and the only perfect explanation is that the antigens in the TB vaccine will be digested by the stomach proteases. So the answer should be 2 only. The answer is D. Okay, let's move on to MCQ number 20. Some RNA molecules called ribozymes can catalyze reactions in a similar way to protein enzymes. So guys, in this MCQ, they are saying that there are some RNA molecules which are called ribozymes. They act like enzymes, which means they can catalyze reactions in a similar way to protein enzymes. Most of these ribozymes have other RNA molecules as their substrates and catalyze reactions that break specific sugar phosphate bonds in the substrate molecule. So what they're saying is that ribozymes are the enzymes that can have RNA as their substrates, right? And ribozymes can break down these RNA molecules by breaking down specific sugar phosphate bonds. Okay, which statements about these ribozymes are correct? Hydrogen, ionic and disulfide bonds will be involved in the ribozyme structure. Guys, this statement is wrong, because as you all know that disulfide bonds, ionic bonds are formed in the protein molecule and not the RNA. As you all know that in RNA molecules, disulfide bonds are never formed, ionic bonds are never formed. Hydrogen bonds are formed in the RNA molecule, but ionic and disulfide bonds cannot be formed. The active site of the ribozyme is formed from a specific sequence of nucleotides. Yes guys, this is true. As we all know that in a protein enzyme, the specific site is this active site, sorry. As we all know that in a protein enzyme, the active site is formed by the specific sequence of amino acids. So in case of RNA enzymes, that is the ribozyme, the active site will be formed by a specific sequence of nucleotides, because RNA contain nucleotides. This statement is true. The statement 2 is true. Ribozymes, which are enzymes, can form because RNA can have a specific secondary and tertiary structure. Yes guys, this is true. RNA can have a secondary and tertiary structure because RNA can have hydrogen bonding. And RNA are forming ribozymes, and obviously ribozymes will have a 3D shape just like protein enzymes. So ribozymes will have a tertiary structure and secondary structure as well. So 2 and 3 options are right. The answer is D. Let's move on to MCQ number 19. The diagram shows the amino acids in the polypeptide. Okay guys, so we can see the polypeptide. It has 4 types of amino acids: polar basic, polar acidic, polar and non-polar. Okay. An enzyme catalyzes the hydrolysis of bonds between the polar basic amino acids and non-polar amino acids. So they say that there is an enzyme which is catalyzing or breaking down or hydrolyzing the bonds between which 2 amino acids? The polar basic amino acid, which is the box, and the non-polar amino acid, which is the circle. Right. How many peptides, chains of amino acids, will be formed as the result of hydrolysis of this polypeptide? So they are asking, if this polypeptide is hydrolyzed by this enzyme, how many chains of amino acids or peptides will be formed? So guys, let's figure it out. What we need to do is we need to break or hydrolyze between the circular amino acid and a box, because box is polar basic and circular is non-polar. So the first point where the break will occur is between valine and histidine, because this is the bond between a polar basic and a non-polar. Histidine is polar basic and valine is non-polar. Okay, let's look for the second point. Yes, this is the second site where the break will occur. Okay, what about the third one? We need to see a box and a circle. Okay, this is the third point where the bond will break by the enzyme. Okay, guys, this is the fourth point where the bond will break, and this is the fifth point where the bond will break. This is the sixth point where the bond will break. Okay, right. Any other point that we have missed? No, I don't think so. Okay, so how many peptides do we get? Peptides are chains of amino acids. So this is the one peptide that we get, chain of amino acids. This is another peptide. So two peptides up till now. Then this one is the third peptide. This one is the fourth peptide up till now. This one is the fifth peptide. Guys, this is the amino acid, so we will ignore this. And this is the sixth peptide. So how many peptides do we get? 6. The answer is B. Let's move on to MCQ number 18. The graphs show various distance measurements taken from metaphase of mitosis onwards. The graphs are to scale when compared to one another. Okay, so guys, these are the measurements of the distance taken during the mitosis right after metaphase or metaphase onwards. So guys, if we draw a diagram showing metaphase, so what happens during metaphase? During metaphase, what happens is that the chromosomes align at the equator of the spindle. Chromosomes align at the equator of the spindle like this. Okay, so this is metaphase, and if we talk about different distances at metaphase, so what could be different distances? One distance could be the distance between the centromere. As you all know guys, that this is the centromere. So we can say that one distance is the distance between the centromere and the centriole. This is the centriole, or we can say the pole of the cell. So one distance is between the centromere of the chromosome and the centriole or the pole of spindle, or you can also say that pole of the cell. This is one distance. Okay, then another distance is between the poles of the cell or the centrioles, the distance between centrioles, right? So for example, this is distance A, this is distance B, for example. So we can say that A is the distance between centromere and pole of the cell, or the pole of the spindle. The distance B is the distance between two poles, distance between two poles. And what could be another distance? The distance between the chromatids. So guys, if we see that chromatids are joined together right now, there is no distance between the chromatids. The distance between chromatids right now is what? 0. Okay, what happens after metaphase? Yes guys, what happens after metaphase? Anaphase occurs, and there is shortening of microtubules, due to which the centromeres split and the chromatids move towards opposite poles. So the picture in anaphase is that there is shortening of microtubules, and what happens is that the chromatids, they move towards opposite poles. Because of the shortening of microtubules, what happened, that centromeres split and chromatids pull towards opposite poles. So now what happens, guys, that the distance between the centromere and the pole, this distance decreases, right? And eventually it will become zero when the chromatids reach the poles. Okay, what will happen to the distance between the chromatids? Yes, guys, the distance between the chromatids was zero initially. For example, this is distance C, distance between chromatids. So this was initially zero in metaphase. Now in anaphase, this will increase. Okay, what about the distance between the poles? Guys, the distance between the poles of the cell will stay constant. The distance A decreases, the distance C increases, and the distance between the poles, which was the distance B, it stays constant. So guys, over here we can see these three distances that I have mentioned. Initially a distance is constant, and then the distance decreases to zero. So guys, when the distance was constant, it was metaphase, and in anaphase the distance decreases and becomes zero. So this is the distance A actually, which is the distance between the centromere of the chromosome and the centriole or the pole. So eventually what happens, that the centromere or the chromatid, they reach the pole and the distance becomes zero, right? So what is X, guys? X is simply the distance between the centromeres from the poles of the spindle. Okay, let's move on to graph Y. Which distance stays constant throughout in metaphase as well as in anaphase? This is the distance between the poles. Yes, guys, this is distance B. So Y should be the distance between the pole of the spindle. And what about distance Z? Distance Z is basically this distance, which was initially 0, but afterward it was 0 in metaphase because the chromatids were stuck to each other. So the distance between the chromatids was 0, and now in anaphase what happens? The chromatids separate and the distance between the chromatids increases. So Z is the distance between the sister chromatids. The answer is C. Okay, let's move on to MCQ number 17. The eyepiece lens of a microscope can be fitted with eyepiece graticule. So guys, they say that, as you all know, that in a microscope there is a device called eyepiece, where an eyepiece lens is fitted, and through the eyepiece we look at the image on a microscope. So guys, this eyepiece is fitted with eyepiece graticule, which is a scale. Okay, which of these statement about the eyepiece graticules are correct? So guys, they have written 3 statements regarding the eyepiece graticule, and we have to select the correct one. They measure the actual length of the cells in micrometers. Guys, this is a wrong statement, because as you all know that eyepiece graticule scale has arbitrary units or false units. Eyepiece graticule scale does not have true units. That is, eyepiece graticule cannot measure in micrometers or millimeters. The divisions on the eyepiece graticule signify arbitrary units or false units. They help biologists to draw cells with correct proportions. Yes, guys, this is true, because when viewing a specimen, when viewing the specimen using the eyepiece graticule, for example, there is a cell. and if the student wants to draw a cell, so using an eyepiece graticule, what student can do is measure the graticule units. For example, if the graticule units that the student has measured, for example, if these units are 9, if eyepiece graticule units are 9 for length, and for width, if these eyepiece graticule units are 10, for example, then if the student wants to draw this drawing, the student can draw the length for 9 centimeters and height or width for 10 centimeters, right? So actually this scale is not used to measure the actual length of the cell in centimeters or in micrometers. This just gives an idea to the student about the correct proportion. ठीक है, ये जो ग्रेटिक्यूल स्केल है ये एक आइडिया दे देगा. क्या आइडिया देगा ये ग्रेटिक्यूल स्केल? ग्रेटिक्यूल स्केल एक आइडिया देगा कि लेंथ के हिसाब से विड्थ कितनी है. ठीक है ना. अगर लेंथ जो है वो 10 यूनिट्स है और विड्थ 9 यूनिट्स है, तो उसके हिसाब से आप जो है वो ड्रॉ कर सकते हो ऑन द पेपर. So this statement, second statement, is true. They change in size when the objective length is changed from into 10 into 40. Always remember this point, guys, that eyepiece graticule is fitted into eyepiece. इसकी कोई slide नहीं होती. There is no slide for eyepiece graticule, right? So eyepiece graticule जो scale है वो eyepiece में fit हुआ हुआ है. ठीक है. It's not on the slide. अगर हम slide place करें, if eyepiece graticule scale was on a slide and if we put the slide on the stage of the microscope, तो फिर वो scale magnify होता. Magnification is only for something that is on the slide and placed on the stage. So if any scale is on the slide of the microscope, if अगर हम slide use करके वो scale लगाएं, तो सिर्फ वो magnify होगा, eyepiece graticule magnify नहीं होता. The answer is 2 only. The answer is D. Let's move on to MCQ number 16. The diagram shows a protein molecule. 2 long polypeptides each form alpha helices for much of their length, and these twist together into a fiber. At one end, each of these polypeptide chains coil into a globular head. 2 short polypeptide chains bind to each globular head. Let me explain you. Guys, this is a protein molecule. This entire structure is of a protein molecule. So they say that there are 2 chains which are forming alpha helices, and what these chains are doing, that these chains are forming alpha helices, right? There are 2 chains and these chains are intermingling or are being associated, right? And then one of the chain, each chain then what it does, each chain forms a globular head, a ball shaped head. Each chain forms a ball shaped globular head. Okay, so up till now we have discussed 2 polypeptide chains. Now they have mentioned that on the globular head there are 2 short chains which are folding. So these 2 short chains are folding on each globular head. So how many polypeptide chains are there? Yes guys, there are 6 polypeptide chains. Okay, what describes the protein structure of this molecule? So guys, we have to select a statement which is describing the entire protein. You have to select that statement which will describe the entire protein and not just the one single chain. Quaternary structure, because each molecule consists of 6 polypeptides. Yes guys, this is the right answer, because the best explanation or best description for this protein molecule, the entire molecule, is the quaternary structure, because there is more than one polypeptide chain. There are 6 polypeptide chains. If we go and look at the other options, secondary structure, because the long polypeptides form alpha helices. Guys, this is the true statement, but not for the entire protein molecule. Secondary structure is true for the long polypeptides, not for the entire protein structure. We have to talk about the entire protein structure. Tertiary structure, because the alpha helices form a fiber. No, tertiary structure is just for a single polypeptide chain. Tertiary structure, because the heads form globular proteins. No. tertiary structure is only for the single polypeptide chain, right? We have to talk about the entire protein structure, protein molecule. Okay, let's move on to MCQ number 15. This is a tough MCQ. The diagram shows a graduated slide, okay, with divisions of 0.1 millimeter viewed using an eyepiece graticule. Guys, the graduated slide means graduated slide is basically a slide with stage scale. Slide with stage scale. This is a graduated slide, and when you place the slide, a scale appears on the field of view. So this is the scale which is found on the slide, known as the stage scale, and this scale over here at the bottom is the eyepiece graticule, which is fitted into an eyepiece. Okay, pollen grains were grown in a sugar solution and viewed using the eyepiece graticule. Okay, so pollen grains were grown, and the growing pollen grains were observed using an eyepiece graticule. These pollen grains are put into sugar solution. Diagram 1 shows the pollen grains at the start, and diagram 2 shows the pollen grains after 4 hours. What is the growth rate of the pollen tube? So guys, what we have to do is we have to calculate the growth rate of the pollen tube. What is meant by the growth rate of the pollen tube? We need to find that what is the change in length, or what is the increase in length in the pollen tube per hour. That is actually the growth rate. So first we need to measure the change in the length of the pollen tube, or we have to measure the growth of the pollen tube. So how can we do that? We need to select a pollen grain which is in line with the scale. Always remember that, select a pollen grain which is in line with the scale. So which pollen grain is in line with the scale? This pollen grain is in line with the scale. Also this one. Both are same. So these pollen grains are in line with the scale. Let's measure. So how many graticule units? If we measure by putting the scale, how many graticule units? 1, 2, 3, 4, 5, 6, 7 graticule units over here. So initial length of the pollen grain is 7 graticule units. Let's look at the final length of the pollen grain. Okay, so how many graticule units? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, right. So 16 graticule units. Let's make it 17, so that we get a good answer. We can approximate the values. इतना ज़्यादा फर्क नहीं पड़ता. Let's take it 17. So the change in length is 10 graticule units. But this change in length is in graticule units, and we need to find the change in length in micrometer or millimeter. Let's find the change in length in micrometer. Guys, from the above diagram where calibration is given, we can calculate the value of the change in length of pollen grains in micrometers. Okay, so from the above information, we can see that the information says that this scale, which is the stage scale, has one division of 0.1 millimeter, that is equal to 100 micrometers. So from this from these two scales that are matched with each other, we can see that 50 graticule units are equal to 100 micrometers. So what is the change in length of the pollen tube? It's 10 graticule units. So what is the change in length of the pollen tube in micrometers? Yes guys, the answer is 50 into 100 into 10 divided by 50. So what will be the answer? 100 divided by 5 is 20 micrometer. So this change in length is 20 micrometer in 4 hours, and we have to find the rate. So to find the rate, we will divide 20 by 4. The answer will be 5 micrometer per hour. The answer is A. Let's move on to MCQ number 14. What explains why the left and the right sides of the heart, they contract simultaneously? So guys, we have to choose a right statement to explain why the left and the right sides of the heart, they contract together. Okay, both atria have a sinoatrial node. No guys, only right atrium has a sinoatrial node. Both sides of the heart are supplied by the same coronary artery. Guys, the contraction depends on the electrical activity. So as we all know that when the sinoatrial node contracts and relaxes, it releases the wave of electrical excitation in the right atrium, and this wave of electrical excitation travels from the right atrium to the left atrium, from the right side of the heart to the left side of the heart, because there is no barrier to the electrical activity between the right and left sides of the heart. So the answer should be D, that there is no barrier to the electrical excitation between the two sides of the heart. That's why the electrical impulse or the electrical excitation can straight away go from the right side to the left side, or from the right atrium to the right, from the right atrium to the left atrium. Purkinje tissue links the two sides of the heart. No guys, Purkinje tissue links the atria with the ventricles together with the AVN. It does not link the two sides of the heart. The answer is D. Okay, let's move on to MCQ number 13. A short piece of DNA, 15 base pairs long, was analyzed to find the number of nucleotide bases in each of the polynucleotide strands. Some of the results are shown. So guys, they say that there's a short piece of DNA which is 15 base pairs long. 15 base pairs mean that there are a total of 15 pairs. It means that each strand contains how many nucleotides? Each strand contains 15 nucleotides. So strand 1 has 15 nucleotides and strand 2 has 15 nucleotides, because the DNA is 15 base pairs long. And what do we have to find? How many nucleotides containing guanine were present in strand 1? So guys, we have to find out how many molecules, how many nucleotides containing guanine were present in the strand 1. So how do we figure that out? It's simple. You should know that the thymine in strand 2 should be equal to the adenine in strand 1. Why is that so? Because of the complementary base pairing. The number of thymine in the strand 2 should be equal to the number of adenine in strand 1, and the number of thymine in the strand 1 should be equal to the adenine in strand 2. Same goes for cytosine and guanine. Okay guys, we have 4 adenine in strand 1, 6 cytosine in strand 1. We have to find the guanine, that is X. Thymine are 3 in strand 1, and total nucleotides in strand 1 are 15. So to find guanine, what we can do is simply we can subtract 15. We can subtract 6, 4 and 3 from 15. The answer will be 2. The answer is A. Let's move on to the MCQ number 12. The diagram shows a model to demonstrate mass flow. Okay, so this is a model which is demonstrating mass flow. In a plant, what are the structures W, X, Y and Z, and what is the direction of the flow of solution along W? Okay, so guys, we can see that Y has a concentrated sucrose solution. It means that Y is a source, right? Because Y has a concentrated sucrose solution, and as you all know that at source, in sieve tube, at source, sucrose is loaded and there is a high concentration of sucrose, or we can say that sucrose is very high in concentration. Sucrose is very high in concentration, and the solution of sucrose becomes concentrated. So this is sieve tube at source, or we can say this is the source. Okay, so as you all know that because of the concentrated sucrose solution, the water potential at Y will be lower. So the water will enter the Y by osmosis. From where, guys? The water potential is lower. So the water enters Y from where? Normally, when the sucrose is loaded into the source, into the sieve tube at source, what happens? The water potential falls and the water from the neighboring xylem vessels come into the Y. So this means that X is xylem. Okay. Then what is Y? Y is a source. And over here, what is an example of source? The leaves. Okay. So guys, what is Z? Z, you can see that Z has a dilute sucrose solution. So this means that Z is the sink, because at sink sucrose is being unloaded. Z is definitely sieve tube at sink, because from the sieve tube at sink, what happens? The sucrose is unloaded. As a result, there is dilute sucrose solution. Sucrose is lower in concentration, which means that there is a higher water potential at Z. So water moves out of the sieve tube, and this water can go into xylem. Okay. So guys, as we can see that Z is the sieve tube at sink. So this means that Z could be the roots. Okay. So we can see that Y is the sieve tube at source and Z is the sieve tube at sink. So there will be mass flow from Y to Z along W, and W is the phloem. So W is phloem over here, and the movement is from Y to Z. The answer is B. Let's move on to MCQ number 11. The table shows the observations made by a student about 3 different cell types, P, Q and R, seen in transverse section of a plant stem using a light microscope. So guys, what student has done is that the student observed a transverse section of a plant stem under the microscope. The student took the transverse or cross section or horizontal section of the plant stem and observed it under the microscope. The student observed 3 types of cells and made some observations. So we need to recognize that what are these cells. Okay. So we can see that the P has no cell contents. It means it's definitely xylem vessel element. One more thing which will confirm that P is a xylem vessel element is that if we look at the ratio, this is the ratio of cell wall width to the whole cell width. What is meant by this ratio? What we do is that we measure the width of the cell wall and divide it with the whole cell width, right? So this ratio is the highest. 1:10 is higher than 1:20. This means that the higher ratio of cell wall width to whole cell width means that cell walls are very thick. When cell walls will be very, very thick, the width of the cell wall will be higher as compared to the whole cell. So this ratio will be highest. If we talk about Q and R, what is Q and what is R? Regular pattern of circles in some cells. So guys, if you look at the transverse section of the stem and you see some cells with regular patterns of circles like this, for example, like this. So it means that you are looking at the sieve tube or sieve tube element, because the regular pattern of circles are basically the sieve pores. So Q is the phloem sieve tube element. If we talk about R, guys, R is companion cell because there is granular appearance of all the cells. If we view the companion cells in cross section under the light microscope, we see granular appearance, and this granular appearance is due to the mitochondria. The answer is D. Let's move on to MCQ number 10. The graph shows how the distance between the sister chromatids, curve P, and the distance between chromatids and centrioles, curve Q, changes during part of the mitosis. Guys, we have just discussed another MCQ like this. Okay, which statement could be correct? So guys, they are talking about mitosis. Mitosis is M phase. So they are asking that which statement is correct, or which statement could be correct. T2V represents interphase. No guys, it cannot represent interphase, because they are talking about mitosis M phase, and interphase is not a part of M phase. T2W represents metaphase. T2W represents metaphase. Okay. So guys, curve Q represents what? The distance between chromatids and centrioles. And as we all know that during metaphase, the distance between the chromatids and the centrioles stays constant. You can look at the diagram that we discussed previously. So guys, in this diagram, we can see the distance A, that is the distance between the centromeres and the centriole or the pole. It stays constant during metaphase. So it's correct that T2W represents metaphase, because during this time, the distance that stays constant is the distance between the chromatids and the centrioles, or the distance between centromere and the poles. This is the same thing. And after metaphase, what happens? During anaphase, what happens? That the chromatids are pulled to the centrioles, or the chromatids are pulled towards the opposite poles or centrioles. So the distance increases. So the distance decreases. Distance decreases and becomes 0. Okay. W to X represents anaphase. Yes guys, this is true. From W to X, we see a decrease in the distance between the chromatids and the centrioles, because the chromatids are moving towards the opposite poles or centrioles, so distance is decreasing. And also, curve P represents the distance between sister chromatids. As we all know that before anaphase, the sister chromatids are stuck together, they are attached together, so distance is 0. And then as the anaphase begins, so from W to X, which is anaphase, what happens? The distance between the sister chromatids increases. The answer is 2 and 3B. Let's move on to MCQ number 9. The photomicrograph shows a section through the lung tissue. Which structures are present in the photomicrograph? Guys, always remember this point, that because this is a lung tissue, so lung tissue cannot contain trachea. As you all know that trachea bifurcates or branches into two bronchi before the bronchus enters into the lung. You all know that first trachea divides into two bronchi, and each bronchus enters into the lung. So the lung tissue cannot contain the trachea, which makes the answer straight away A. Okay, I would like to label the structures and help you identify these structures in this diagram. Okay guys, so if we talk about this photomicrograph is less magnified, because it can be further magnified to even view the smaller structures. Okay, so guys, what are these that we can see? Over here, this structure that I am just highlighting is the vein. How do I know that this is the vein? Guys, this is a large vein, because you can see that this structure has thin walls and wider lumen, and this is irregular in shape. So guys, this is a large vein. If we talk about this structure, so guys, this is definitely an artery. This is definitely a large artery, because it has thicker walls and comparatively narrower lumen. This is also a large artery with thick walls. Guys, if we talk about these structures, so these are definitely bronchi. Why? Because you can see the cartilages, you can see the irregular blocks of cartilage. Irregular blocks of cartilage. There is no C-shaped cartilage. Obviously, C-shaped cartilage is found in the trachea, and trachea cannot be in the lung tissue. This is also, yes guys, this is also a cartilage. This is also a bronchus with irregular cartilage. So in this photomicrograph, we can see the bronchus, we can see the vein, we can see the artery, but we can never see the trachea. Guys, if I zoom in this diagram, so what are these? These air spaces that you see. Let me make it clear for you. Guys, these air spaces that you see are the alveoli. Right? Okay. Can you see this structure? I'm just highlighting. Guys, this is a bronchiole. And how do you recognize the bronchiole? By the wavy epithelium and thin walls. What is this? Guys, this could be an artery, but this is a small artery, which is equal to the size of the bronchiole. Right? Guys, this is another bronchiole, and these are the alveoli. Okay. These again are the arteries, but small arteries. Okay. Let's move on to MCQ number 8. The diagram shows a metabolic pathway that generates an important hormone. So guys, there's a metabolic pathway which is generating an important hormone. And how is this hormone generated? There's a reactant which is converted to intermediate X, then intermediate X is converted to intermediate Y, intermediate Y is converted to hormone, and there are 3 enzymes which are responsible, or there are 3 enzymes which are taking part in this pathway to form hormone. A metabolic poison is able to slow the production of the hormone by inhibiting one or more enzymes in the pathway. So they are saying that there is a metabolic poison that can slow down the production of this hormone by inhibiting one or more enzymes in this pathway. The effect of adding this poison is a large increase in the amount of the reactant. So guys, if there's a large increase in the amount of reactant, this means that this reaction has slowed down, right? If reactant is building up, this means that this reaction, the rate of this reaction has decreased. It means the poison is blocking the enzyme 1. Okay. And a decrease in the amount of intermediate X. Obviously, if the enzyme 1 is blocked by the poison and the rate of reaction falls, then the intermediate X will decrease in concentration. Okay. There's a slight increase in the amount of intermediate Y. Okay guys, if intermediate X falls, so intermediate Y should also fall. That's because if there's less intermediate X, so less intermediate X will be converted into less intermediate Y. So intermediate Y should fall, but intermediate Y is rising. What could be the reason for this? The poison also blocks enzyme 3. That's why intermediate Y is rising. Normally, if the enzyme 3 was not blocked, the intermediate Y should also decrease because less intermediate X is converted to intermediate Y. But intermediate Y is rising. That's because enzyme 3 is also blocked by the poison. So the rate of this reaction is slow. And there's a large decrease in the amount of hormone. So by this information, we can be sure that enzyme 1 and enzyme 3 are blocked by the poison. Which enzymes is metabolic poison targeting? It is targeting enzyme 1 and enzyme 3. The answer is D. Let's move on to MCQ number 7. The graph below shows a calibration curve for the absorbance of different concentrations of glucose in a solution. Okay guys, so this is the graph of absorbance, and this absorbance is measured by the colorimeter, and this absorbance is according to the different concentrations of glucose. So guys, is glucose a colored solution? No, it's not. So how the glucose can have different absorbance? This is because of the Beta-thalassemia test. As we all know that as the concentration of glucose increases, if you carry out a Beta-thalassemia test, glucose is a reducing sugar, and as the concentration of glucose increases, the color is darker. As you all know that when glucose is in trace amounts, the color of the Benedict's solution is green, and as the concentration of glucose increases, the color of the Benedict's changes from, it becomes darker, it becomes yellow, orange, red, it becomes darker. It does not stay green. Okay, so absorbance will be more. Over here we can see that as the concentration of glucose increases, the absorbance also increases. That's because the color with Benedict's solution is getting darker. Okay, calibration curves can be used to determine the concentration of unknown samples. A student wanted to determine how much sucrose they had in an unknown sample using the figure above. To make their results comparable, they first added sucrase to hydrolyze the sucrose into glucose and fructose. Then they set their colorimeter to the same settings as one used in the calibration curve and tested the unknown sample. The sample had an absorbance of 25%. How much sucrose was present in the unknown solution? So guys, if we see the calibration curve, so this is a calibration curve, and at 25% absorbance, there is how much concentration of glucose? This is 200 milligrams per deciliter, right? And in this MCQ, they are asking about how much sucrose was present in an unknown sample. So what do you think? How much sucrose will be there in the unknown sample? Okay. So over here, from the 25% absorbance, we see that the concentration of glucose over here is 200 milligrams per liter. Okay. So if we talk about sucrose, when hydrolyzed is broken down into glucose plus fructose. And we can see that the ratio is 1:1 between sucrose and glucose. Even if we draw a chemical equation, this will be C12H22O11. This is sucrose. When hydrolyzed, forms C6H12O6. This is glucose plus C6H12O6. This is glucose. So always remember this point, that the ratio is 1:1, 1 mole to 1 mole. So the concentration of glucose will be same as the concentration of sucrose. If glucose is 200 milligrams per liter, same will be the concentration of sucrose, that is 200 milligrams per liter. The answer is B. Okay, let's move on to MCQ number 6. The graph shows the dissociation curves for hemoglobin at two different partial pressures of carbon dioxide. So guys, this is a graph which is showing the hemoglobin dissociation curves at two different partial pressures of oxygen. So the curve on the right is at the higher partial pressure of oxygen, and the curve at the left is at the low partial pressure of carbon dioxide. At which positions on the graph A, B, C or D is the concentration of hemoglobinic acid lowest in the red blood cells? So guys, what is hemoglobinic acid? Hemoglobinic acid is HHB. So if we recall the entire pathway for the formation of HHB, we can see that carbon dioxide combines with water to form carbonic acid. This reaction is catalyzed by which enzyme? Carbonic anhydrase. And then carbonic acid dissociates into hydrogen ions and hydrogen carbonate ions, right? Okay. What happens then? There is oxyhemoglobin which combines with hydrogen ions to form hemoglobinic acid, that is HHB, and oxygen is released from the oxyhemoglobin. HbO8 is oxyhemoglobin. So these are the pathways. Okay. So we have to select a point where hemoglobinic acid concentration is the lowest. Always remember this point, that guys, if partial pressure of CO2 is low, what will happen? Less carbonic acid will be produced, less hydrogen ions will be produced, and less HHB will be produced. So always remember that if there is a low PCO2, there will be low HHB, and if there is a high PCO2, then there is high HHB. Always remember this point. We can figure out this by the equation. If there is low CO2, less carbonic acid will be produced, less hydrogen ions will be produced, and less hemoglobinic acid will be produced. And if there is more carbon dioxide, more carbonic acid is produced, more hydrogen ions are produced, and more HHB are produced. Okay. If we talk about oxygen, so guys, if we have more oxygen, so this reaction will shift towards the left side, and we will have less HHB. So remember this point, that if we have high partial pressure of O2, there will be less HHB, and if we have less partial pressure of oxygen, there will be more HHB. So we have to select a point where HHB is lowest. So for HHB to be lowest, partial pressure of carbon dioxide should be low for the HHB to be lowest, and partial pressure of oxygen should be high for HHB to be lowest. So where is the partial pressure of carbon dioxide lower? On which curve? On the curve that is on the left side. This is the curve where partial pressure of carbon dioxide is low, and C is the point where partial pressure of oxygen is highest. So the answer is C. Okay, let's move on to MCQ number 5. Which row is correct for the mean blood pressure in different parts of the human circulatory system? Guys, always remember this point, that blood flows from high pressure to low pressure. So guys, as we all know, that blood is flowing from high pressure to low pressure, and if you look at these structures, artery in arm, vein in arm, capillary in arm, and right atrium, we know that blood first flows from artery in the arm to the capillary in the arm, and then from the capillary in the arm to the vein in the arm, and from the vein in the arm the blood goes into the right atrium, right? This is the route of the blood. The blood flows from the artery in the arm to the capillary in the arm, and from the capillary in the arm to the vein in the arm, and from the vein into the right atrium, because all the blood from the body is returned to the right atrium via the veins. So because blood is flowing from high pressure to low pressure, the pressure in the artery should be highest, because the blood is flowing from the artery to the capillary. Capillary will have a blood pressure lower than artery but higher than vein, because blood has to flow from capillary to vein. And the right atrium will have the least pressure, because the blood has to move from the vein into the right atrium. So the answer is A. Let's move on to MCQ number 4. 4 nucleotides A, B, C and D each consists of 3 phosphate groups, a nitrogenous base and a pentose sugar. Characteristics of the base and sugar components before they are joined to each nucleotide are shown in the table. Which nucleotide could pair with an adenine base during DNA replication? Okay guys, so they are saying that there are 4 nucleotides and each consists of how many phosphate groups? 3 phosphate groups, a nitrogenous base and a pentose sugar. And over here there are characteristics of individual components of the nucleotides before they are joined to form the nucleotide. We have to select a nucleotide that could pair with an adenine, which means that the nucleotide must contain what? A thymine base. Why not uracil? Because guys, they are talking about DNA replication. So we need to select from these options, we need to select a nucleotide that will pair with adenine during the DNA replication. This means that the nucleotide must contain deoxyribose, because it should be a deoxyribonucleotide, because they are talking about DNA replication. So the nucleotide should contain deoxyribose sugar, and because the nucleotide has to pair with an adenine base, the nucleotide should contain thymine. So the ring structure of the nitrogenous base that the nucleotide should contain must be single ring. Why? Because thymine is a pyrimidine with a single ring structure. So the ring structure of the nitrogenous base which will pair with adenine should be single. What about the ratio of carbon to oxygen atoms in the pentose sugar? Guys, pentose sugar over here is deoxyribose. If we talk about deoxyribose, the formula of the deoxyribose is C5H10O4. However, this is very unusual because as you all know that monosaccharides, as you all know that general formula of monosaccharides is is CH2ON. This is the general formula of monosaccharides. So according to the general formula, the formula should be C5H10O5. Why? N is the number of carbon atoms, and in pentose sugar there are 5 carbon atoms, as you all know. So the general formula, according to the general formula, the formula should be C5H10O5. But the formula of deoxyribose is C5H10O4. So according to the general formula for the pentose sugars, the formula should be C5H10O5, but the deoxyribose has a formula C5H10O4 because of one less oxygen. As we see in the name of deoxyribose, there is deoxy, so there is one oxygen atom less as compared to the normal pentose. That's why there are 5 carbons and 4 oxygens. So carbon to oxygen ratio is 5:4. So in the pentose sugar, the ratio of carbon to oxygen atoms is 5:4. Let's say if we talk about ribose, so ribose will have this formula. In case of ribose, the carbon to oxygen ratio will be 1:1. 5:5, which will be cancelled out to 1:1 in case of ribose. Okay, MCQ number 3. The jellyfish Turritopsis dohrnii is described as being immortal. If T. dohrnii is not eaten by predators or diseased, it seems to be able to live forever. There is no way to determine the biological age of the T. dohrnii individual. Which feature of the cells in the T. dohrnii could explain these observations? So guys, they say that jellyfish is immortal, it does not age or it does not die until and unless it is killed by the predators or is killed by the disease. So guys, how can an individual does not age? How come an individual does not age or does not die naturally? This is because of the telomeres. If the cells of an organism contain an enzyme called telomerase, so the telomeres will keep on topping up the telomeres, and the telomeres will not shorten. What happens during DNA replication is that the telomeres shorten because the DNA polymerase does not copy the DNA till the end. So some length of the telomere is lost and the telomeres shorten. So as the telomeres shorten, eventually the telomeres finish, and when the cells divide, then so the vital genes are lost. Normally, how do we age? If we talk about us, so how do we age? We age because as the cells divide, telomeres shorten and eventually all the telomeres are lost. So when the cells still divide, now instead of telomeres, because telomeres are not there, vital genes are lost. And when our vital genes are lost, we lose many abilities. For example, the hair turn white, the skin get wrinkles, we lose stamina, and the bones get weak, and the body gets weak. So that's all because of the loss of the telomeres and the loss of the vital genes. If in any organism telomeres are not shortened, or there's an enzyme telomerase which can top up the telomeres, or if there is an ability to restore the telomeres, then aging will not occur and death will not occur until and unless eaten by the predators, or if a if the organism catches a disease. So which feature of the cells in the T. Dohrnii could explain these observations? An ability to restore telomeres to their original length is the answer. Right? Very long G phases. No, this could not be the answer. Long G phases does not help to prevent aging or death. Very short S phase. No. Fewer chromosomes than other eukaryotic organisms. No. MCQ number 2. Which statements about a peptide bond are correct? It joins two monomers which are always identical to each other. No, guys. Peptide bond joins two amino acids, and two amino acids could be different. They are not always identical. Okay, it contains 4 different atoms. This is true. Well, this is a misleading statement, and this statement confuses the students. But let me tell you that if you look at the peptide bond, so peptide bond is associated with how many atoms? 4 atoms, which are 4 different atoms. These are oxygen, carbon, nitrogen, and hydrogen. So this is true, and the orange one over here is the peptide bond. So peptide bond is associated with 4 atoms, or we can say peptide bond contains 4 different atoms. This is true. Okay, it can be broken down by the addition of water at room temperature. No, guys, peptide bond can only be broken by the enzyme, and or peptide bond can only be broken down by the enzyme or by heating it with dilute hydrochloric acid. Right? Peptide bond cannot be broken down by the addition of water at room temperature. It is important in the primary structure of the proteins. Yes, as we all know that primary structure is the sequence of amino acids in a polypeptide, and the amino acids are joined in a particular sequence with the help of the peptide bonds. So peptide bonds are important in the primary structure. Peptide bonds hold the primary structure of protein. The answer should be D, 2 and 4. Let's move on to MCQ number 1. Which statements about mitochondria, chloroplasts, or prokaryotes are correct? So guys, we need to take the correct statements regarding the mitochondria, chloroplasts, or prokaryotes. Mitochondria and chloroplasts have a fully permeable inner membrane. Guys, this is wrong. Inner membrane and outer membrane both are partially permeable. Membranes are not fully permeable because membranes are made up of phospholipid bilayer. Phospholipid bilayer is permeable to non-polar molecules and impermeable to polar molecules, so membranes are never fully permeable. Prokaryotes and chloroplasts have 70s ribosomes that are sites for translation and polypeptide synthesis. Guys, this is true. As we all know that prokaryotes and chloroplasts, they both have 70s ribosomes, and these are the sites of translation and polypeptide synthesis. Prokaryotes and mitochondria have an outer membrane and an inner folded membrane where ATP synthesis occurs. Guys, this is wrong. Mitochondria has an outer membrane and an inner membrane, but prokaryotes do not have outer and inner membranes. Prokaryotes only have a single membrane. Prokaryotes and mitochondria have circular DNA. Yes, they do, where genes coding for information are located. This is true statement. The answer is 2 and 4 only. The answer is D. So guys, we are done with the difficult MCQs. I hope you found these MCQs challenging and you understood these MCQs very well. Guys, for further videos, subscribe to my YouTube channel. Thank you so much for watching. Allah Hafiz everyone.